Hello, potential viewers. Welcome to my channel. Bret Weinstein, the one sort of infamous evolutionary biologist and former Evergreen College professor, recently went on to the Joe Rogan Experience again. And at this time, he finally shared his story from his graduate student years, when he discovered that the main source of lab mice of US pharmaceutical labs and companies had a severe selection problem in its lab mice breeding process. This was definitely not the first time Mr. Weinstein shared his story. If I recall correctly, he mentioned that he shared the story with many people of various media outlets many times before, but none of the attempts gained any traction. The first time I heard of this was on Eric Weinstein's podcast, The Portal, which I highly recommend, by the way. Eric was Bret's brother, and they both had their perspective on the story. It has a decent audience, but Joe Rogan Experience was definitely the biggest platform on which this story was shared up to today. What I find a bit frustrating is the way that Bret told his story. And on both platforms, namely Eric Weinstein's podcast, The Portal, and Joe Rogan Experience, this issue was especially obvious. Bret's storytelling style is very nuanced and well-reasoned, and he has a habit of providing a lot of context when he makes a point. This would be just fine if the story was not overly complex with scientific backgrounds, and he was not talking to someone who is either an equal scientific mind like his brother Eric, with whom he seemed to just glance over some important contextual points while focusing on others, or someone who has a habit of interjecting their own premature understandings and drug-related anecdotes like Joe Rogan. So in this video, I will attempt to retell the story in a relatively layman manner and provide as much context in simple terms as possible. And hopefully you will understand it better if you did not fully understand it or get to hear it in the first place. If you felt a chill running down your spine by the time you finished this video, just like I did when I first heard the story, that would mean I achieved what I set out to do in this video. So first of all, let me provide some points of key contextual information. First is the mice and rats are widely used in pharmaceutical researches. I think this is not an obscure knowledge. Scientists and researchers use them because they are fairly cheap and easy to breed, and they are physiologically close to human enough so that they can be used as models for preliminary drug tests. If a drug is effective on humans, it is expected to be effective on mice and rats, and any side effects that are possible on humans are expected to manifest somehow on mice and rats as well. Second point is the idea of antagonistic pleiotropy. This is a term describing the evolutionary phenomenon of evolutionary traits in all living organisms where they are beneficial in short term yet harmful in the long term. A lot of these traits are preserved in our evolution because in most cases, an individual organism will have offsprings to pass on their genes and/or already died before the harmful effects of the traits started their impact. An example would be the testosterone level in men. Higher testosterone levels will help men build up more muscles and perhaps higher bone density according to some new studies during younger years and thus become advantageous in competition for mates. Yet they may lead to higher risk in prostate cancer in later years. And the third point is the notion and functionality of telomeres. To quote Wikipedia, which has a much better explanation than I can drum up. A telomere is a region of repetitive nucleotide sequences at each end of a chromosome, which protects the end of the chromosome from deterioration or from fusion with neighboring chromosomes. End quote. And in layman's terms, it is a repeated sequence of DNA at the end of each cell's chromosome. Each time a cell splits through mitosis, the telomere gets shorter, and when it gets to a certain length, the cell can no longer split, thus constituting as a mechanism for limiting how many times cells within an individual organism can split. This limit on cell split was called the Hayflick limit, discovered by biologist Leonard Hayflick. This, as Bret Weinstein found out, is theorized by a lot of cancer researchers to be a mechanism of cancer prevention, since the more a cell splits, the higher the chance that it contracts a mutation in the process and turns cancerous. On the other hand, telomeres can be augmented by the activity of a special and relatively restricted kind of protein in one's body called telomere enzyme, which is mostly active only in stem cells. and cancer cells often possess the ability of exploiting telomere enzyme to allow themselves to clone indefinitely. Now, with these three key points of contextual information, let's recap the core of Bret Weinstein's story. During his graduate student years, because of his interest in the mechanism of evolution rather than the mere phenomenology of it, which was relatively rare in the field at the time due to the lack of observational tools on cellular levels, he attended a lecture by a cancer researcher in his class. During this lecture, the cancer researcher introduced the theory that the limited length of telomeres having something to do with some sort of cancer prevention mechanism within individual organisms, that the telomeres serve as the limiters of cell split, which will terminate the lineage of a cell after too many splits in order to prevent the offsprings of the cell from turning cancerous. This lecture provided a shocking insight to Bret Weinstein. In his own words, it struck him like a bolt of lightning. As he is very familiar with the theory of antagonistic pleiotropy, that evolution prefers evolutionary traits that are beneficial to an organism in the short term, even though these traits may become harmful in the long run. As explained before, the reason was that by the time the harmful effects take place, the organisms will have already produced offsprings, and thus fulfilled its purpose of spreading genes. And there was one unfound factor. When considering the theory of antagonistic pleiotropy, this is the biological phenomenon of senescence, or aging in layman's terms. And aging is essentially a process where more and more cells inside of an organism reaching their limit and in turn stop reproducing. This causes the damages to the organism's tissues and organs to go unrepaired and become weaker over time and eventually die. It has long been theorized that aging itself is in a way beneficial to the organism in a way, comparing to some other outcome. Yet no one was very clear on what the other outcome was, because in theory, unlimited cell splits means our tissues and organs can be repaired forever, and we do not have to die of old age, right? Thus, the cancer prevention theory provided us with the elegantly simple answer that the risk introduced by cell splits is cancer. And unlimited cell splits means cancer will surely come into the picture and kill the organism. This introduces us to the paradox of life and death of an organism. That an organism, even disregarding all other complicating factors that will affect its mortality, will either die of old age or die of cancer. And the process of natural selection over the millennium mostly found the sort of balance between these two factors in the form of life expectancy. Organisms become vigorous enough to live past sexual maturity, to live as many offsprings as possible, yet not too long, lest cancer kills them. Being a pretty well-read student and struck with this incredible insight, Bret Weinstein became interested in another peculiar phenomenon, that lab rats and lab mice have weirdly long telomeres, 10 times the length of human telomeres, for the species' short life expectancy. From there, he started suspecting that the lab rats and mice that are being systematically bred and selected in order to serve as drug test models might be subject to a process of natural selection that led to this phenomenon, where rats and mice in the wild do not have as long telomeres as their lab-bred counterparts. So Bret Weinstein contacted the then-known expert in the field, now Nobel laureate Carol Greider, and asked her lab to confirm this theory. And later, the theory was confirmed. Wild rats and mice do not have as long telomere strings as their lab-bred counterparts. What is also confirmed through the conversation was that, when left untouched, a lot of lab mice and rats will eventually die of cancer. Also, when the labs order lab mice and rats from European suppliers, the lab rats and mice do not have telomeres as long as the ones from American suppliers. Or should I say, American supplier. Bret Weinstein found out that the absolute majority of the lab mice and lab rats that are bred inside of the US, they come from a single place, Jackson Lab in Maine, also known as Jack's Lab, JAX. Thus, it stands to reason that something is very wrong with the breeding process inside of Jackson Lab, for the lab mice and rats coming from this organization to all have extremely long telomeres. And due to it being the absolute biggest supplier of lab mice and rats being the de facto model organism for almost all drug tests, it would be obvious that the absolute majority of the pharmaceutical drugs that are researched and produced in the US have been tested on these lab mice and rats. But what is the concern here, one might ask? To that, the answer will be the effect of the extreme length of telomeres is severe enough to compromise the drug test results. As stated before, the length of telomeres limits the times cells can split. Also, the means in which an organism recovers from damages to its tissues and organs, either from outside wounds or, say, toxins and toxic side effects from experimental drugs, is to have healthy cells replicate to replace the wounded and dead cells. And if more healthy cells are able to split and replicate, the quicker and better the recovery. Thus, the longer the telomeres one organism has, the stronger its ability to heal the damages inside its body and stay young. But also, as stated before, it will become more susceptible to cancer. What this means is that when pharmaceutical companies do drug tests on these lab rats and mice with extremely long telomeres, they are subject to two huge biasing factors without even knowing and understanding it. That is, test subjects they are operating on are much more resilient against tissue and organ damaging toxins and side effects from their drug because of their enhanced ability to heal and recover. Yet, at the same time, these test subjects are also much more prone to cancer, making drugs that are designed to combat cancer seem way less effective as they would otherwise be on a human. The latter carries an implication that we may have had some effective treatment for cancer already, if not for the test subjects already being extra susceptible to cancer. The loss cannot be calculated or even estimated, since the potential cancer cure never made it past the lab rat testing. The much more serious implication is the former. Therefore, we have absolutely no idea how long pharmaceutical companies have been testing on these lab mice and rats when it comes to the toxicology of their product. These rats and mice, being extra resilient to drug toxins and side effects, are in turn being extra permitting of drugs that can cause long-term tissue and organ damage in humans. Yes, there are drug trials with human subjects. However, it is extremely difficult to measure slow-building long-term tissue and organ damage in human drug trials. For this kind of trials, the common practice is to test on lab mice and rats with large doses in order to observe the manifest effects. Yet with the aforementioned reason, the results are very often clouded. And we may have already seen the aftermath of such widespread skewed test results. There were already several drugs being recalled for causing heart failure. As predicted by Bret, the long-term result of these misbred little lab rats and mice is this. We do not know how many drugs that can deal severe long-term tissue damage are on the market right now, but there is an easy tell. The failures of organs that are more susceptible to tissue damage will surely climb, heart failure being one of them, due to the fact that heart cells are less able to split and heal the tissue when damaged. And it is not very hard to deduce that organs with similar conditions of low self-repairability are also prone to the same harm. Filtration membrane in kidneys, eyes, to name a few examples. Needless to say, this should be a DuPont Teflon dark water level of a scandal. But as Bret Weinstein frustratedly pointed out, no major media outlet was willing to touch this story. Everyone knows rats are not good models, some said, and some just stopped connecting with him after reaching out to some quote-unquote experts in the field. Even Carol Greider, an expert in the field, treated the discovery as some sort of a fun anecdote to bring up during her Nobel acceptance speech. Now that the story made it on Joe Rogan Experience, the largest podcast in the world, I sincerely hope, though I am not holding my breath for it, that the story will gain much more traction than before. That will be all for my video today. I will leave the stories from respective podcasts in the link below. Please share the story as far and wide as possible.